we finished the break. Now we continue. There's some questions regarding the Western blot. I think I should clarify. So the question is, why do you need to do that? Because you can see the protein after Komatsu blue. Right? Why do you need to do Western blot? When we do Western blot, we usually use whole cell extract. Like I give you an example. I gave you an example of liver cell before and after hormone stimulation. So we run the whole protein produced in one cell onto the gel. They are like, you know, we have 20,000 genes in our genome. So there are thousands of different proteins in the protein extract. You can't see your protein. So this is to detect the presence of your protein among a very, very heterogeneous mixture in your protein extract that you extract from liver cells, in my example. Okay, so you're not purifying the protein and use Western blot for detection. You see what I'm trying to say here? So it's very heterogeneous. Okay. Okay, so we continue, and I have to be a bit faster so that we can finish all the material. You can go home and digest and come to ask me questions next time. So this is called isoelectric focusing, a technique that we're going to talk now. And it's a kind of equilibrium electrophoresis system, very similar to isopycnic centrifugation in terms of concept. So before we talk about isoelectric focusing, first of all, you have to understand that proteins are polypeptide chains, right? So it contains of a chain, a polymer of amino acids. We have 20 amino acids generally. And some of the amino acids are acidic in nature. Some of them are alkaline in nature. So acidic amino acid, aspartic acid, glutamic acids, at high pH, these side chains will be negatively charged. But if you put these amino acids side chain at low pH, it will assume a structure like this. So it's a COOH that has no net charge. Okay, so this amino acid side chain, therefore, will change its charge according to the surrounding pH. And the same is true for lysine, arginine, histidine. So at high pH, lysine, arginine, and histidine has no net charge. But at low pH, it will be positively charged. So the whole picture is that the whole protein at high pH will be more negative charged. And at low pH, it will be more positively charged. Is that as simple as that, right? So we're talking about the same protein that has, say, 5 of these negative charge protein and 5 of this positive charge protein, okay? And each one of these has a different pKa. So it's a dynamic system that we're talking about. So you put this protein at pH 10, then this protein is negatively charged, right? Because at high pH, the lysine will have a structure like this, NH2, but the aspartic acid will have COO minus, so it's negative charge. And then you slightly and progressively change the pH of the solution surrounding this protein. So pH 10, pH 9.5, pH 9, pH 8.5, and slowly change that. Then what you will see is that this protein will progressively from negative charge to become less and less negatively charged. and then you change the pH to a certain pH. At that particular pH, the protein will have no net charge at all. Does it make any sense? Okay. So this thinking process, you can do also in this region here. So the same protein you put at a low pH environment, pH 4, for example. This protein is positively charged. And then you slightly and progressively change the pH from low pH going upward, then this positive charge protein will become less and less positive until it becomes no net charge. Okay, so the same protein, either going in this direction or in this direction, will meet in the middle, and this is the isoelectric point of the protein, where it has no net charge. Okay, so this is the whole principle of the idea. And all you need to do is to generate a pH gradient across the gel. And of course, we can do that because we can buy solutions. They're called ampholyte solutions. If you put ampholyte solutions into the gel, then you apply a charge, electric field across the gel, it will generate a pH gradient for you. And you can put the protein anywhere in the gel, it doesn't matter. Once the pH gradient is generated, you put the protein here in the low pH region, it will move to its isoelectric point. And if you put the protein in this region, it will move to the isoelectric point. Okay, so it really doesn't matter. But of course, in the laboratory, we will always load from one side, the protein, but it really doesn't matter. So this is the whole idea. So if you can generate a stable pH gradient, the same protein here is positively charged, will move, and it's moved towards the side to become less and less positive, and become no net charge at this point. And this protein here, negative charge, and move, and again, no net charge here. So this is the isoelectric point. You can therefore focus this protein into this band by using its own property. Okay, so this is how it's being done. So we usually use tube gels. You can set up a tube gel by including an ampholyte solution that you can purchase from company. So it will generate a stable pH from pH 9 to pH 3. And then afterwards, you load your sample on top, and then you resolve the protein according to the pI of the protein in the gel. So that's simple to understand, right? So the concept now is that usually nobody goes through the trouble of doing PI, isoelectric focusing, because I mean it's a lot more difficult compared to SDS to run. But if you combine the two techniques together, it's called two-dimensional gel, and it is very, very, very powerful. And in fact, I'm sure you have heard of the term proteomics, and the whole idea is based on 2D gels. Okay. If you can separate your protein using IEF on one direction, and then you put this gel on top of a separating gel here, and separate all these proteins in second dimension. So one side is PI of the protein, and the other side is the molecular weight of the protein. You can therefore separate a very, very heterogeneous mixture of proteins. And then if you do it today, do it tomorrow, do it the week after, do it in my lab, do it in your lab using the same apparatus and the same condition, then you can reproduce the same result. And you can actually take a picture of this 2D gel, and then you can catalog that. And nowadays, we can actually take each one of this dot out, and we can sequence this protein. Or you can use mass spec to test the molecular weight of this protein to precisely to the last digit. And then you can use it for identification of this protein. You can imagine that I use the same example as the Western blot. Okay, so I stimulate liver cell with a hormone like glucagon. The other side, I don't stimulate with glucagon, with no hormone. And I run a 2D gel of the liver extract. And then I scan this gel with a densitometer. And then I scan the gel with and without, what I mean is with the hormone stimulation, without the hormone stimulation. You can therefore take a look and compare each of these dots. You can see how many proteins are being stimulated by the hormone in the whole cell, and how many proteins are being repressed or silenced by the hormone. Because each of these proteins, you can identify them using mass spec or using sequencing technique. So it's a very, very powerful technique, and the whole area of proteomics therefore was raised, developed based on this 2D gel. Okay, so this is a physical way to separate the protein. You can also think about any other non-physical way in solution form, in chromatographic form, to separate the protein in 2 dimensions and then do things. Okay. But the whole idea is that there's a very powerful means to systematically separate a heterogeneous mixture of protein, and then you can use it for detection to understand biological cellular processes. Okay, so the last topic of my lecture is chromatography. One of the lab is based on cell size exclusion chromatography. So separation of protein based on size. Okay, and the protein gel, the SDS page lab, is also kind of depending on the affinity chromatography technique for the purification of the recombinant protein. So 2 labs related to this topic. And the idea of chromatography is to use a column, a physical column. Of course, there are non-physical way to separate using the very similar idea like paper chromatography. But we only talk about column chromatography in my lecture. So it's to separate molecules in by 2 phases. One is called a stationary phase, which is immobilized. So you can see, imagine that in a column, you can pack the column with a matrix, and that is an immobilized part of the separation. And the other one is a mobile phase. It could be liquid or gaseous, but in our lab, we only talk about liquid chromatography. Okay, so you will constantly allowing a liquid to flow through the gel matrix. And after you load your protein mixture into the gel, basically you're asking the protein to take side. I like the matrix more, or I like the liquid more. So if the protein like the matrix more, it will retain more in the gel matrix. And if the protein like the liquid more, it will go out from the column earlier. Right? So basically you're allowing separation of protein based on how they like to be in the stationary phase or in the mobile phase. So basically, what you're talking about is different distribution coefficient of the proteins in the stationary phase versus the mobile phase. Okay, so how much they'd like to in these two different phases. So the concept is simple, right? So you have a column that you pack with the matrix, and then you load your analyte. In this example, you have analyte A and analyte B. And analyte B like the matrix more, analyte A like the solution more. So they can be separated. So when you constantly allow, after you load your sample, you allow a constant flow of liquid down this column. A, because it likes liquid more, so it will go faster. And then it will come out earlier. So this is the retention time. And then if you can collect fractions, then you can collect this fraction. Analyte B, like the matrix more, it will come out later. So you can have, therefore it has a different retention time, and then you can collect this fraction. And based on that, these two proteins originally in the same sample is now being separated. They're being purified into two different fractions. Okay, so this is the whole idea of chromatography. And we have gel filtration or size exclusion chromatography. And this technique is separation based on size. We have ion exchange chromatography. It could be either cation or anion exchange. And this is based on charge of the protein. And then you have affinity chromatography, and this is based on shape of the protein. You can imagine if you have an antibodies against an antigen, you can use that antibody, conjugate that to the matrix, and use that to purify your protein, the antigen of interest. Okay. So this is your lab, basically. So you have packed a column with the beads, with the gel, and then afterwards you load your protein. And in gel permeation chromatography, the smaller size protein can access the volume, the holes inside the gel matrix, because they're smaller. And the gel has a definite size of holes, channels in the gel matrix. Therefore, smaller protein can enter the gel and access that region. Bigger protein are excluded from the gel matrix. They can't get in at all. So they will just come out right away. So this is the principle for separating proteins of different sizes. Okay, so basically, it's the same technique that we're talking about, same diagram that we talked about earlier on in this diagram here. But now, this diagram shows you that you are, after you load your protein sample in the gel and continue run the gel, after you load your protein in the column, continue run the column, continuous dripping of the solution in the column, then the large protein will come out first because it is being excluded from the gel matrix. Smaller protein has a much longer path length because they can enter into the gel matrix and will come out later. And this is the elution profile. You can collect fractions here, combine fractions to separate and purify these two different proteins, as shown here. And in your lab, you'll be given a column. Each one of you will be given a Cephadax gel column. We will pack the column for you. We will equilibrate the column for you. You'll be given a sample that contains 3 different analytes. It contains blue dextran, molecular weight of 2 million, very big molecular weight. So this molecule will be excluded from the gel. Will not enter the gel matrix at all because it's so big. It contains cytochrome C, which is a small protein, molecular weight of about 12 kilodalton. And it also contains cobalt chloride, molecular weight very small. And then you are going to separate these three molecules using your Cevadex gel, each one of you running a column. And you will tell me the distribution coefficient, KD, of cytochrome C in the end of the experiment, based on the data that you collected. So the idea is that the total volume of the column is the solution volume plus total volume is the solution volume plus the gel volume, right? The gel has volume, the gel matrix has volume, but it contains three regions. The gel volume, the volume of solution inside the gel beads, and the volume of solution outside the gel. Okay, so total volume of the column is VO outside, V in inside, and V gel, the gel volume. You don't have to think about VG at all, but you will have to think about VI and VO. Okay, so when you think about blue dextran with such a big molecular weight, then it's totally excluded from the gel matrix. So the elution volume of blue dextran is the same as VO. It's the outside volume, right? And then for cobalt chloride, it has such a small molecular weight, it have total access to the column, then the elution volume of cobalt chloride is therefore the total solution volume of the column. And it is VO, outside volume, plus the inside volume. And the range at which you can separate proteins is somewhere between VO and VO plus VI. So this is the only solution range. That this is the only volume range that you can separate your protein. Okay, so based on your laboratory data, you will tell me what is the VO by looking at blue dextran. You will tell me what is VO plus VI by looking at the elution volume of cobalt chloride. And you will tell me the distribution coefficient of cytochrome C by looking at the elution volume of cytochrome C. And the equation is like this: VE equals to VO plus KD times VI. And if you know VO, and if you know VI from here, and if you have the elution volume of your protein of interest, then you can therefore calculate KD from this equation. It's just a simple mathematical equation. Okay, so okay, so these gel matrix you can buy from different company. Again, we usually buy from Pharmacia or Biorec, and they have different gel matrix that can separate molecule of different sizes. So, for example, Cevadex G10 cross-linked dextran and fractionate up to 7 very small molecules. Cevadex G200 from 5,000 to 800,000. You can fractionate You can like allowing molecule going into the gel matrix in this range of molecule, from 5,000 to 800 kilodalton. And BioX similar, two different matrix you can purchase, P2 and P300. And there are of course much more in the market that one can buy, depending on what size molecule that you like to fractionate. Okay, so we finished talking about the gel exclusion chromatography. The next one is ion exchange. It's basically a similar idea. I'm sure you can understand like this. So the only thing that you need to know is that the gel matrix could be negatively charged, then it's separate positive charged protein. It could be positively charged, and it's separate negative charged proteins. So imagine that in this case, you have a positively charged beads, and then you load your sample. So all neutral positive charged proteins will come out first because they don't interact with the gel beads, right? And then you elute your protein using a gradient of salt. Okay, the small net negative charge protein will come out first at low salt concentrations. And then the large net positive charge protein will come out later in higher salt concentrations. And the way that you can visualize what is happening is that, so this is the gel matrix here, positively charged. And then you have three different proteins here, 1 negative, 2 negative, 3 negative. At low salt medium, low salt concentrations, they all interact with it. At medium salt, the 1 negative charge will come out first. And then at higher salt, the 2 negative charge will come out, and slowly the 3 negative charge will come out later, just like that. So you can separate based on the charge density of your protein. Again, you can buy these from different companies. So you can have anion exchanger and cation exchanger, and they are based on different charge of the matrix. So you can have, in this case, diethylamino ethyl group. It's a weak anion exchanger, pH range 3.6, and it's used for separating proteins. And for the cation exchanger, you can have carboxymethyl as a side chain that is also weak, and fractionation range 7 to 10 for the separation of proteins, but opposite charge, of course. Okay, so the last technique is called affinity chromatography. And affinity chromatography, of course, is the method of choice because it specifically interacts with your protein, right? So you can purify your protein using one column. And this is what you will do in your lab. And you have your column packed with a matrix that is conjugated with a structure. And then you load your sample, and any other thing that do not interact with that structure will come out in the flow through. You wash your column several times to make sure that there are no contaminated protein in the gel in the column anymore. And then you can elute your protein of interest. But for this technique, the difficult part is the elution, because your protein interact strongly and specifically with the structure that is conjugated to the column. So the difficult part is how to elute your protein, but not to the nature of protein. And basically, the two desorption methods, one is specific, and the other one is non-specific. Most of the time, I prefer myself to use specific method. So one can use a higher concentration of the same ligand that you conjugate to the column to elute your protein. So your protein conjugated, sorry, your ligand conjugated column, your protein interact with the ligand. And if you, in the solution, provide a higher concentrate of this ligand, then the protein will bind to your ligand in solution and come out from the column instead. In that way, you can preserve most of the time the structure of the protein. For non-specific desorption method, you can change the pH. Changing pH oftentimes change conformations. You can change the ionic strength. Sometimes it works, lower salt, higher salt. You can use a denaturing agent, such as urea, or an organic solvent. And all these methods are non-specific. Okay, so it's not specific to the interaction. And the specific desorption is by using a specific ligand that you put into solution that interact with your protein, competing with your protein already bound onto the column, and you can dissolve. Okay, so I'll stop here today. Next time we will talk about some sample questions, exam format, and then there will be a tutorial afterwards. Okay, and everything will be recorded. But I hope, I mean, face-to-face teaching is so much more fun compared to Zoom teaching. I hate Zoom teaching, so I enjoy teaching you guys. Good. I'll see you next week. This